All right. So up to this point in the previous class, we talked about central dogma and talked about some of the different processes that that constitute central dogma: DNA replication, transcription, and translation. Today, we're going to be talking about DNA replication. And historically, we've been talking about these different subjects against the backdrop of some sort of disease. With DNA replication, there's not really any disease that that'll help us explore that, because as you can imagine, if you do not have DNA replication, you do not have life. And so we're going to be looking at chemical and biological warfare that tries to leverage DNA replication and attack it. And so historically, there have been lots of different chemical and biological weapons that date back thousands of years ago, whether it was the Spartans that would ignite and toss sulfur, or the Chinese Sung Dynasty that would use arson that was aerosolized, or the Mongols taking plague-infected bodies and launching them over walls. And so one of the more recent examples of that is mustard gas. The proper chemical name is up there to the top, but what's noteworthy about mustard gas is that it's a powerful alkylating agent. What that means is it adds alkyl chains to different molecules in our body, and it dates back to the 1800s and was most widely used during World War I, after which there was an international treaty to outlaw chemical and biological weapons. But the sad reality is that it still often is used even today. The noteworthy part about mustard gas is that it's a vesicant, it's a blistering agent. And what's important to know is, as you can imagine, it being developed back in the 1800s, it's easily made and dispersed. It can easily be stored, and it's oftentimes difficult to treat, decontaminate, and protect against. And so some of the symptoms of mustard gas is erythema, which is inflammation of our mucous membranes. In addition to that is conjunctivitis, which is inflammation of our conjunctiva, the membrane within our eye, and it causes this blistering and burning. The blistering affects our nasal mucosal area, which causes irritation, sneezing, and severe nosebleeds. As you can imagine, as it affects our oral respiratory mucous membranes, it can lead to severe coughing and sore throats and severe vomiting. It is so lethal that it has an LD50 of about 1.5 grams. As we talked about before, LD50 is the dose at which 50% of the population would die. And if it is in the lungs, it only requires about 10 milligrams. And so because it also affects the surface area of our skin, it can also have severe effects at only 0.5 gram per minute over a cubic meter of space. And so it's incredibly effective, incredibly lethal. That's one example of a chemical agent. The biological agent that we're going to try and use to better understand DNA replication is anthrax. It's also historically was referred to as wool sorter's disease, during the industrial age, when people would be shearing wool from sheep. It oftentimes comes from the bacteria Bacillus anthracis, and it's often found in the soil and can be infected in sheep. And when they're shearing the wool from sheep, if they have cuts on their hands or knuckles, it can come from that. And so the people who were sorting wool oftentimes were infected with it. And so that's why it was historically called wool sorter's disease. It can primarily infect large vertebrae like sheep, as the historical name implies. And what's important to know is that it will undergo this change to form these spores that will protect it in unfavorable growth conditions. And so when anthrax is in its spore form, regardless of its high humidity, low humidity, high temperature, or low temperature, that helps protect against it in those harsh conditions. And those spores are what oftentimes are used when it's used as a biological agent. When I was preparing for this lecture, whenever I think of anthrax, I think of the Anthrax Scare that happened in 2001. And I quickly realized that that might have been a year before a lot of you were born. But what ended up happening was a few weeks after 9/11, there were letters sent to people throughout several people throughout Congress in which there were anthrax spores in these letters, and it led to this huge scare where people were afraid to open their letters. And so oftentimes for people, it's weird to say this, my age and older, there's a really tight connection with anthrax to that anthrax scare, where it was actually a molecular biologist who wanted more money for anthrax vaccine research that ended up sending it to members of Congress in an act of biological warfare domestically. So it's a favorite of weapon makers because, again, when it's in its spore form, it's protected and it can survive for years. It's easily grown and it can be easily dispersed. As an example, you open a letter and you can quickly become infected. And it has a really high mortality rate. But what's also unfortunately elegant about this form of biological warfare, it's non-transmissible, meaning that it can be incredibly targeted to attack one single person, but it's not contagious or cannot be transmitted to other people. So there's a couple different forms of anthrax. There's cutaneous anthrax. It's only usually possible through a cut or abrasion. So when talking about the wool sorters who'd be infected, oftentimes their knuckles or hands would be cut sorting through this wool, and it can only the spores can only enter in through an abrasion or some sort of entry point into the skin. Now it's oftentimes transmitted through interaction with an infected animal, and it's usually not lethal when there's antibiotic treatment. The symptoms, as you can look at this image here, it's oftentimes characterized by a raised bump with an ulcer, and we talked about what that was before. That's this pus-filled infection with a necrotic center around it. We've talked about apoptosis being programmed cell death as being really elegant as this coordination between cells. Necrosis is just this unregulated cell death that is far less elegant, but more of like a sledgehammer, just killing everything around it. And it also is characterized by swollen lymph nodes locally in that area. As we described before with the secondary lymphoid organs, like the lymph node, we have lymph nodes all throughout our body. And so it's the local lymph nodes that become swollen around the infection site. Inhalation anthrax is where it becomes far more deadly. As you can imagine, it is characterized by inhaling the anthrax spores. And so the infection begins in the lungs, and in a matter of days, the mortality rate is usually about 100%, unless there's antibiotic treatment, which is usually ciprofloxacin, or nicknamed Cipro, by which after that treatment, the mortality rate goes down far less, or the mortality rate is much lower. The lethal dose is about 10,000 spores. And so as you can imagine, when this is a microscopic agent, it's easy to deliver 10,000 spores in a very small delivery agent. And so oftentimes the symptoms begin with upper respiratory infection, which is typically unremarkable. There's oftentimes followed by improvement, but then after that you get dyspnea, which is difficulty breathing, diaphoresis, which is profuse sweating, not just normal sweating, profuse sweating, even more sweating than when I ran here when I was late last time, and myalgia, which is described as muscle fatigue or muscle pain. And it will eventually lead to septic shock. That's a systemic infection that leads to shock. We talked about this before with anaphylactic shock, but shock is usually the drop in blood pressure that's so severe that you can't actually no longer perfuse blood through your organs, and it'll lead to organ failure. And so this is the lethality of anthrax is really important, particularly with inhalation. So the reason why we're mentioning these two chemical or biological agents is that mustard gas, it kills by inhibiting our DNA replication, and anthrax is treated by antibiotics or other compounds that inhibit the bacteria's replication. And so we're going to try and discuss DNA replication both in mammalian or eukaryotic cells as well as in prokaryotic cells, to try and better understand mustard gas and anthrax. Yes. Yes, I believe 9 people were killed and 15 were infected but treated. And so 10,000 spores is again, this is it's microscopic, so that's not, it can fit in a regular size envelope. And so it is, unfortunately, it is incredibly effective. Are there any other questions? Yes. It is pretty easy to detect. Going back to our, I think it was protein biochemistry slide, there are lateral chromatography, lateral flow immunochromatography that you can use to detect anthrax. And so for a long time, the US Postal Service used those tests on a lot of letters that were going to Congress for a long time as a result of this. Are there any other questions? Yes. Yeah, that's a good question. I think domestically, I think there's a tighter restriction on people who are using anthrax, because again, this was a molecular biologist that actually worked for the government. And so I think there's a tighter regulation on who can use it and how they're using it. I do not think that same degree of regulation exists abroad, but I can only speak to, I think, what's been clear as a result of this anthrax scare. Any other questions? All right. So we talked about before what in a previous pre-lecture, we talked about the different nucleotide bases and the hydrogen bonding that occurs between these base pairs that holds them together, and that the hydrogen bonding contributes to these complementary nucleotides, where we have adenine that binds to thymine and cytosine that binds to guanine. And what's important is that, as you can tell in this image, that it's mediated because there's complementary or matching hydrogen bonding between A and T and C and G. Between adenine and thymine, we have 2 hydrogen bonds, and guanine and cytosine, we have 3 hydrogen bonds. And so in addition to that, the paired chains, the paired complementary chains, are what is anti-parallel. We described in the previous lecture how DNA, so we described how polypeptide chains have directionality, and that also DNA strands have a directionality as well. It goes from the 5' to 3' end, but if you look on the complementary strand, it goes in the opposite direction. And so that's what we mean, they're anti-parallel. They have opposite directionality, but they're complementary to one another. And replication, this complementary nature is leveraged when trying to make copies, or trying to use, well, again, what we call the template strand, the strand that's being used during DNA replication to make the subsequent copy. And so the enzyme that is used during DNA replication is DNA polymerase. And DNA polymerase can only do so much during DNA replication. So the reasons why it can only carry out some functions, DNA polymerase itself is unable to separate or melt the DNA strands. And so it's easy. I want to put a pin here and say it's important to know that when we say melting with the DNA strands, it's not like a thermal heat melting, but rather just the process of separating the DNA strands. And so DNA polymerase is unable to do that on its own, and it requires help to make that happen. Another problem for DNA polymerase is that it can only elongate a pre-existing strand, meaning when we talked about DNA replication at a high level before, we talked about how there is an RNA primer that adjoins to the template strand. And so it requires something there to begin elongating the strand. It can't initiate though. So that's why it needs the RNA primer. It gives it something to work with, for lack of a better phrase. And so the two strands in the DNA complex are an opposite polarity, like we talked about with 5' to 3', but this is important. DNA polymerase can only elongate in that 5' to 3' direction. All right. So the natural question as a result of this is, where does DNA replication begin? And so the sites where DNA replication begins, we call those origins of replication or replication origins. And so the replication origins, the sites where DNA replication begins, is recognized by, for lack of a better name for them, origin binding proteins. And so we're going to try and explore this again, looking at eukaryotic versus prokaryotic DNA replication. With prokaryotic DNA replication, these replication origin sites contain unique DNA segments that have these short repeats that are called consensus sequences. And there's usually only one consensus sequence, that there's one single replication of origin site for prokaryotes. But with eukaryotic DNA replication, we do not have these consensus sequences, and we have far more replication origin sites, tens of thousands. And so that these sites are less well understood among prokaryotes in mammalian DNA replication, but in prokaryotic cells, it's these consensus sequences where DNA replication begins. Now, that's one way in which they're different. One way in which both mammalian and prokaryotic DNA replication is similar is that these replication origin sites, they're really rich in and they're AT-rich sequences. Now, thinking about the hydrogen bonding, can anyone venture guess why AT-rich sequences might be the sites of DNA replication? Yeah, that's exactly right. Because it's 2 versus 3, you can imagine it's 33% less work for them to do to try and begin DNA replication at those replication origin sites. And so looking at consensus sequence, looking at regions of DNA across different organisms, the different prokaryotic organisms, you can see that these consensus sequences, when highlighted here, are these regions that are rich in adenine and thymine. So having both prokaryotic and mammalian replication side by side, you can appreciate how on the left, we have one single site, one single origin of replication on the left, which is the consensus sequence. Whereas on the right, the origin is not a consensus sequence, but rather these different sites of replication origins that happen. And we have what we call, this is important to note, so we have the original DNA strand, we call that the parent strand. And so as replication is beginning on the right here, looking at the mammalian example, we have these new DNA strands that are replicating, and we call those daughter strands. And so this is happening at different sites throughout the DNA. And so again, it's oftentimes tens of thousands of sites where DNA replication can begin, whereas for prokaryotes, it's usually a single site, a single origin of replication. And so what will end up happening as the DNA strands are elongating, it begins to form these, what we call them, replication bubbles. And over time, the two will fuse together, and the sites where, when looking at B on the right part for mammalian cells, as the daughter strands elongate, there's the site where the sites at the end, we typically call them replication forks, as you can see labeled to the right. And over time, these daughter strands will fuse together, and you'll have a fully developed copied daughter strand. For the prokaryotic example, because it's a circular structure for the DNA, what will end up happening is that the daughter strand the DNA polymerase will continue to go through around both sides, and it'll eventually create two separate loops that then separate and create copied DNA. What's important here, what I want to try and repeat and make clear again, is that prokaryotic cells usually have a single site for a single origin of replication site that's defined by the consensus sequence. Mammalian or eukaryotic cells do not have that consensus sequence, but rather we have many different origin replication sites. And how those sites are characterized, we're still trying to understand that now. But what's noteworthy is that we have far more than prokaryotic cells. So we have our sites where DNA replication is beginning, and we know that DNA polymerase itself cannot separate. We talked about the problems of DNA polymerase itself cannot separate. And so we have the initiation that's mediated by initiator proteins. Initiator proteins themselves will bind to the origin sites, and this is important, may separate the strands. And we're going to distinguish that between, again, prokaryotic versus eukaryotic. So in prokaryotic cells, the initiator a key initiator protein is called DNA. DNA is capable of separating the DNA strands. In eukaryotic cells, the initiator protein is called the origin replication complex. It's this hexamer protein complex that's characterized by ORC1 through 6. And so in mammalian cells, ORC does not separate the strands. So ORC does not melt the DNA strands themselves. Whereas in prokaryotic cells, DNA, that initiator protein can. And so in mammalian cells, what we use is there's a protein called helicase that does the separating or the melting. And so helicase, after ORC binds to and holds the DNA strand, helicase will come in and separate out the protein and melt it. Yes. Yeah. Oh, that's a good question. You can ignore that. This was pulled from a paper that did the crystallography to define these proteins, and so it's part D and part E of that figure. So DNA A is prokaryotic. The ORC complex, or ORC, is eukaryotic. So this is the initiator protein that we have. And so what's important to know is that it does not separate strands, whereas DNA A can. What does the separating, or what does the melting in mammalian cells is helicase. And so this process, this is a process that does require energy, excuse me. Are there any questions so far? Use this as an opportunity to drink some water. Are there any questions? Again, I love dumb questions. Yes. Yeah. Can you say that one more time? I couldn't hear very well. Yes. Duplex DNA is just double-stranded DNA, or the double helix. And so what's important to know is that helicase in mammalian cells is what melts the DNA. And so, yes, Liam. So it is, it's coming from ATP. And so typically when we describe, whenever we describe an enzyme that requires energy, it's oftentimes mediated by ATP. So in prokaryotic versus eukaryotic cells, there are different helicases. Javi, do you have a question?
Oh yeah, I was just gonna say if DNA A separate strands in prokaryotic, why do they compete?
That's a great question. You took what I was going to say here. So there's because DNA replication is so important, there can oftentimes be redundancies to help make sure an important process is occurring. And so it's unsurprising that in prokaryotic cells that despite them having a smaller and despite them having a smaller genome, they, it's not surprising that they might have a degree of redundancy where their initiator protein can separate, but they still have something, a helicase that can help mediate that melting of the DNA. And so in prokaryotic cells, their helicase is DNA-B. You're seeing kind of like a simplicity in their nomenclature, the nomenclature that's been developed for them. So DNA-B is a prokaryotic helicase. And in mammalian cells, we have a protein that's, the mammalian helicase is mini chromosome maintenance complex, or MCM. And so it's another multimeric complex that helps to unwind or melt the DNA that's important for replication. So as we described, helicase separates the DNA, but as you can imagine, once that DNA is separated, if replication is not happening yet at that site, there's kind of there's a thermodynamic instability. If they're just sitting separated, and if nothing is stabilizing them, they'll want to go back and re-anneal to form the double-stranded DNA complex. So what we have are these single-stranded binding proteins that help bind to single-stranded DNA after helicase melts them and stabilizes them in a single-stranded conformation. And so it helps stop them from re-annealing together so that replication can continue forward. And if you imagine, I wanted to try and use this example, but then I realized I might be aging myself again. When you are separating, so DNA is in a double helix, and when you're separating something that's twisted up like that, what you can get is something called supercoiling. And so the way I'm going to age myself is when you have landlines or phones that have these, you can envision it as DNA as a phone cord on a landline phone. And if you were to be able to pull them apart, you can imagine that that separation is going to create lots of extra coiling further down on the DNA strand or on the phone cord. And so there are proteins that help stabilize the DNA at those distant sites so that supercoiling isn't happening. And so gyrase is a class of proteins that helps to eliminate that supercoiling and help unwind the protein a bit so that supercoiling doesn't happen. And so to try and to take a second to try and help map out what we've said so far, we have the sites of replication. We call them replication origins or origins of replication. Initiator proteins are what come in to initiate replication at those sites, at those replication origins. Our mammalian cells, our initiator proteins can't melt the DNA, and so that's why we have helicase to do that. Single-stranded binding proteins, SSBs, help to stabilize it once it's been separated, and gyrases help to make sure that supercoiling is not happening downstream. So we have a couple examples of proteins that help address that supercoiling problem. One is called a type 1 topoisomerase. The way this works is type 1 topoisomerase is what they'll do is they'll create a single break, or you can think of it as like a single cut or nick, that allows the remaining intact DNA, double-stranded DNA to twist and help stabilize under the torsion of DNA strands separating. And so over time, what will happen is that after DNA replication is occurring, these nicks will be, for lack of a better phrase, repaired or fixed. And so this is a process that does not have any net energy expenditure. This is a pretty simple process that happens in both eukaryotic and prokaryotic cells. And so this is what the structure of topoisomerase 1 with the DNA helix, double helix within it. Yes. Yes, and so that's Yeah, yes, they're both types of DNA gyrases. Type 2 topoisomerase. Oh, yes, you have a question? Yeah, exactly. And so it's not changing the actual bases. What's happening is that there are, if you think about the structure of DNA, we have our bases, we have in DNA, the deoxyribose sugar, and you also have the phosphate group. And so what's happening is that there are cuts being made at different parts. Because if you remember I think if I have, yeah, I'll go back to, so when we go back to the structure of DNA, going back further than I should, we have this phosphate group that joins between different base pairs throughout the DNA sequence. And so what's happening is that there's these phosphodiester bonds between these base pairs with their deoxyribose sugar attached to them. And so the cuts are happening here, and then helping to help bridge them back together. Does that make sense? This is going to make you dizzy on the recording. If you watch this again, I'm sorry. All right. Type 2 topoisomerase. So with the type 1, there's a nick that's repaired. But what ends up happening here with type 2 topoisomerase is that again, it's an isomerase. It's an enzyme. So the substrate here is a point where the DNA crosses one another here. And so what's going to end up happening is that there's going to be a cut that happens on one side of where they cross, and then using energy, once it's completely broken, it'll be passed through. Type 2 topoisomerase is mediating this. The broken segment will be passed through and then resealed on the other side. And so this, again, uses energy. This is an active process. And this is an example of a DNA gyrase. And so, as you can imagine, this is a model of what type 2 topoisomerase is doing. So it'll cut or break the DNA there. It'll open up, the other strand of DNA will pass through, and it'll help rejoin that broken DNA strand. So when we talked about the structure of prokaryotic DNA, it's these circles that it's this circular DNA structure that when it's copied, you have these two rings of DNA, and you can kind of imagine, if you imagine what like the Olympic rings look like, they're kind of like linked together. And so that's what it looks like when there's DNA replication happening in bacteria. And so something needs to happen to help separate them apart, and that's what type 2 topoisomerase is doing. It's actually breaking one strand of the DNA, letting another one pass through, and then fixing that cut so that now you have DNA that's been replicated. And so it helps with the circular chromosome that prokaryotic DNA have. It helps to create, it helps to mediate that separation of a copied DNA. In mammalian cells, our DNA is not just long strings of DNA everywhere, but they're packed tightly in these structures around chromatin in a tight chromosome structure. And so despite us looking at DNA on a screen as like a single double-stranded DNA piece, they exist in tight loops that have been racked together. And so type 2 topoisomerase is really important at helping to copy and then separate out copied DNA that's in these tight compacted structures. Are there any questions there about type 2 topoisomerase or any of isomerases? Yes. That's a great question. They're both used to address supercoiling. And so with type 1, it's making a small cut. It's making a single cut and helping to try and release or reduce the torsion from the unwinding. And with type 2 topoisomerase, what ends up happening is that it helps to relieve it's another mechanism to relieve that supercoiling, but then also is important in helping with the separation of copied DNA as well, or replicated DNA as well. Yeah.
Does that get I guess, because now once there are now still has that, does it have to do the opposite?
It'll automatically coil again, and late at the end of this lecture, we're going to talk about another protein that helps mediate the fixing of those cuts. That helps with ligation, with the rejoining together. Those are great questions. Are there any other questions? Yes. Yeah, so if you think about it, if I'll go back to the model we had. So what's happening here is, as compared to type 1 topoisomerase, here there's there's unfolding and there's movement of DNA strands. And so what ends up happening, it requires energy because you're moving the other DNA strand through into this DNA binding pocket down below. And so it's an active process. Yeah, it's an active process that requires energy, whereas with type 1 topoisomerase, it's not. There's less demand for energy. If that makes sense.
What's causing the fact that there's more energy?
Because with type 2, what you're having is you're actually passing through another DNA strand, whereas with type 1, you're not doing that. Mm-hmm. So as we mentioned before, we have DNA polymerases that will add nucleotides from the 5' to 3' direction. And so there's a few things that are important to help mediate that. Number one is the template. We need the template DNA that's going to be the instructions for creating the complementary strand. We need this is the deoxyribonucleoside triphosphates. And so this serves as both the source for the nucleotides and as an energy source. So if you remember when we talked about what ATP is doing, it's donating phosphate groups that help mediate change. And so here, the phosphate groups on deoxyribonucleoside triphosphates, it's helping to both provide the energy source through the phosphate groups as well as the nucleotides themselves. And the last piece is the primer. We talked about how DNA polymerase cannot initiate; it can only elongate. For transcription, as opposed to during transcription, RNA polymerase, there's not usually a primer. It just during transcription, RNA polymerase is able to initiate and elongate, whereas DNA polymerase can only elongate. And so it needs a primer. It needs something to, for lack of a better phrase, prime the pump to begin transcription or to begin replication. Forgive me. And so we have here what ends up happening, and this is pretty important here. This is pretty similar to some other processes that we've talked about in the past. This is a process by which deoxyribonucleoside triphosphates, or we'll call them DNTPs. That's a much easier way to say it, and it's not a mouthful. DNTPs, what ends up happening is that there are nucleotides that are brought into to the site where replication is happening, where the elongation is occurring, and it's a triphosphate. So these other phosphate groups these inorganic phosphates will leave, and you're going to have the single phosphate group with a sugar and the base. But what's important to know, and we'll talk about this more in a minute, is that this is a process by which if it's not the right base that corresponds to the other nucleotide on the template strand, it'll leave, and other nucleotides, bless you, will be brought in to see if it's the corresponding one. And so it's a process by which there's constantly nucleotides being brought in. We have nucleotides everywhere within the nucleus here. And so they're constantly being brought in to determine whether it's the right matching one based on the thermodynamics of the corresponding nucleotides. And so the DNA polymerase, like we said, it needs a primer. And what helps to provide that primer is called primase. So primase helps to provide these short RNA sequences that serve as the primer. Yes.
Sorry, I have a question on the slide before.
Yes.
In the replication, it's one start with the primer.
So you're asking, is it going to create a circular DNA? So that's only for prokaryotes, not for it's not for eukaryotic cells. And so what's in sites of whenever there's, let me see how to say this properly. For replication in our DNA, like our DNA is tightly compacted and tightly wounded for space conservation. But when replication is happening, what you're having is DNA being pulled apart to mediate that replication. And it's not going to be creating a circular chromatin. So prokaryotic cells have a circular chromatin. We have chromatin that's wrapped around histone proteins. And when replication is happening, it's being opened up to mediate that. But then after, over time, it's being wrapped back around and compacted to mediate to permit chromosomes that can then, this is kind of going all the way back to, I think, the second or third lecture now. There are the mTOCs that help mediate our cytoskeleton to pull apart using the chromosome structure to replicate the whole cell now. And so it's being pulled up, it's being stretched out to mediate replication and then being compacted down so that now the whole chromosome can be pulled apart. Does that make sense? So primase is the enzyme by which we have the short RNA sequences that serve as the primer. And so this is and it requires DNA. And so it creates an RNA primer that corresponds to the template strand of DNA. And it requires DNA. And so this is gonna be a mouthful. It's a DNA, primase is a DNA dependent RNA polymerase. And so it's making RNA, and it requires a template DNA strand for which to make a primer that matches to it. And so again, RNA polymerases are able to undergo transcription without requiring some sort of primer. They can both initiate and elongate, whereas DNA polymerases cannot initiate. They need that RNA primer that primase provides to mediate DNA replication. Yes. Yeah, that's a good question. The only explanation I can think of is a protective mechanism against dysfunctional replication, where there's not in the absence of like a safeguard or a check that requires like a primer to initiate things. If DNA polymerase was able to initiate on its own without the necessity of a primer, I suspect that there might be it's like one less check on replication that might introduce some sort of issue with replication and Yeah, the only thing I can think of is it just being like a safeguard against DNA replication. Because I'm right now I'm racking my brain trying to think of an example in cancer where there's an issue with DNA replication where perhaps DNA polymerase is able to work without needing the initiation. I can't think of a cancer example, but my hunch is that it's a safeguard against dysfunction in replication. So we have two different kinds of DNA polymerases. So in prokaryotes, this DNA polymerase is called DNA polymerase 3. In eukaryotes, it's called delta. So you can either call it DNA Pol 3 in prokaryotes or DNA Pol delta in mammalian cells. And so this is the main polymerase in bacteria. And it consists of, it's a large multimeric protein or multimeric enzyme that consists of multiple peptide subunits. And so what's really important to know is that, as you can imagine, as we've described already, it mediates polymerization or replication in the 5' to 3' direction. And so we've spent all this, I've spent a lot of time trying to repeat that it goes in the 5' to 3' direction. It can also move back in the 3' to 5' direction. Now, but that's not for polymerization. That's for exonuclease activity. As you can imagine, exonuclease activity, what that means is that exo at the ends, what that means is it's trying to proofread the nucleotides to make sure that they're the matching pair. And so if, for example, if we have the incorrect base that's added here, moving from the 5' to 3' direction, what will end up happening is that because we have a guanine that's trying to be paired or matched with a thymine, it's not going to have the right hydrogen bonding to have the strands joined together. And what's going to end up happening is that it's going to have a kink, and that's going to disrupt the ability for DNA polymerase to continue with polymerization. So that change in conformation is actually going to elicit this proofreading, this exonuclease activity, where it'll actually now move back and remove that incorrect base. And to try and again, now that it's removed that base, it's going to try and go back to its polymerization activity and try and mediate adding the right base. And so when I was talking a few slides ago about DNTPs being the source for nucleotides and how there's constantly nucleotides being assessed if they're the right one or the wrong one. When they're the wrong one, DNA polymerase 3 or DNA Pol delta, it'll use its exonuclease activity to remove the wrong nucleotide here. And to now that it's gone back up, now it's going to move in the 5' to 3' direction to try and mediate adding the right nucleotide. Are there any questions there? I want to pause there for questions because I've been talking about it only moves in the 5' to 3'. Now we're talking about how it can move back. Yes. Correct. Yeah, you got it. That's part of it. So like it has two roles. It can act as a polymerase or act as a exonuclease. And so perhaps what will help is it is acting as a polymerase as it's adding nucleotides during elongation. And I talked about how DNTPs are the source of energy and the source for the nucleotides. And what's happening is that we have nucleotides everywhere. So as DNTPs are adding in these nucleotides, if it's the wrong one, DNA polymerase 3 or DNA Pol delta will be able to sense that because of this change or this kink in the conformation because of the wrong pair. And now because the hydrogen bonding isn't matched up. And so now it'll be able to detect that, and then it'll switch roles and now act as a nuclease to remove that wrong base pair. And once it moves back up, now it's now that it's changed its role, it now moves in a different direction. It's now going to move from 3' to 5'. And so once it gets to a point now where the conformation is correct, it can now switch roles. Now it went from exonuclease to a polymerase, and now it'll move in the 5' to 3' direction, helping to mediate the addition of the right nucleotides. And so this is a process that's constantly undergoing, where it's constantly proofreading to make sure things are okay. And if it's not, it's going to switch activities, move in the opposite direction. And once it gets to once it reaches a point where the conformation is correct, it's going to switch back and now be a polymerase and keep going in the 5' to 3'. And so it'll, as I described a moment ago, it'll remove the wrong nucleotides and switch back to its polymerase role and begin polymerization again. So the name exonuclease, it's cleaving the nucleotides at the ends. And what it can also do is it can also act as an endonuclease, where it can cleave them in the middle. So exo at the end, endo in the middle. And the final product is going to be synthesizing the reverse complement of the parent strand. So when I say reverse complement, again, DNA has directionality, the 5' to 3'. So if we were going to read this parent strand, we'd be reading it CTTACAG, and it's going to create the reverse, because it's moving this way now, the reverse complementary strand. And so whenever we're reading DNA, we're going to be reading it from the 5' to 3' direction. And so the DNA polymerase 3 is going to help create the reverse complementary strand. Are there any questions there? I want to pause for a minute. I'm going to drink water. I want to leave time for questions. I want to get at least a couple questions. Yes. Yeah, so it can act as because of its proofreading abilities, there can sometimes be breaks in the DNA, and so it has the ability to assess if there's any wrong nucleotides in the middle, if that makes sense, because there can be breaks that are made by, for example, Topoisomerase 1 that can just make a cut there, and sometimes DNA polymerase 3 is able to go in and assess if they're proofread as an endonuclease. What I want you guys to know, or what I want you guys to focus on, is the two main roles that I've highlighted above, as it being a polymerase and exonuclease. It can function as an endonuclease as well, but what I want you more to focus on is its role as an exonuclease. Does that help? Okay. Yes. Okay. So it is. I want to make sure I understand your question. Is your question? Is it creating? Is your question that is it creating a complementary strand, or is it creating a daughter strand that's 5' to 3', or reading the parent strand 5' to 3'? So what it's doing is it's creating in the 5' to 3', but it's reading here at the 3' to 5'. Does that make sense? So it's what ends up happening is it's creating a new daughter strand in the 5' to 3' direction that's reverse and complementary to the parent strand. All right, and so this is a structure of this is a model structure of what it looks like with DNA polymerase 3 or DNA Pol delta with the with the DNA that's been opened up and single-stranded, and that's been opened up and is mediating the replication of a single-stranded DNA there. Yes.
So is this like DNA polymerase 3 and DNA polymerase delta? Like, of course, they're the same, but are they different in structure because they're parts of like, one prokaryote, one is eukaryote?
That's a good question. I don't think there's. Say that again. Sorry.
Sorry, Rocky, what is this? Like, is this DNA polymerase 3 or is this DNA polymerase delta?
So that's a good question. DNA polymerase 3, to my knowledge, DNA polymerase 3 and DNA pol delta, they're the same enzyme, they're the same structure. It's just different nomenclature depending on what context we're in. If we are in a prokaryotic context, we're going to call it DNA polymerase 3. If we're in mammalian or eukaryotes, we're going to call it DNA polymerase delta. Yes, Cole?
Could the DNA polymerase be used to edit and customize?
To edit and customize. So that goes to that. We have not been historically. We have not been able to customize and edit DNA until the advent of CRISPR Cas9, and so that leverages. I want to make sure we've talked about enough stuff to talk about CRISPR Cas9 yet. It leverages natural cuts in the DNA, and that you have this. You basically have like a primer RNA that you can serves as your primer RNA, and that you can also use to help write the DNA as you'd want it to be. But that's the only way in which we've been able to customize DNA. Historically now only in cells. The clinical trial I talked about in the very first lecture for sickle cell disease is the first US clinical trial where we've been able to use CRISPR Cas9 in people to repair the hemoglobin beta gene as it should be. Does that help answer your question? It's still the ability to write DNA is fairly new, and there's a lot of ethical bodies that are trying to regulate that to make sure it's not abused, because as I understand it, there are scientists in other countries that have went ahead and just started using it on unborn babies and to try and see if we can customize children, for lack of a better phrase. And so that's not what DNA polymerase is doing here, or that's not, we can't do that with DNA polymerase. We have to use other resources to try and do that. And so what's important to know is that as replication is happening, and as you can imagine, as the DNA polymerase is going through and replicating and creating the copy strand, it's really important to try and make sure that the DNA is the double-stranded DNA is not re-annealing. And so before replication begins, we have those single-stranded protein binding proteins that help stabilize the single-stranded DNA. And so there's a function of DNA polymerase, not a chemical function, but rather a physical function that helps to try and keep the the DNA in a clamp down. It helps, the clamping is done by DNA polymerase to try and make sure that it's not letting go of the DNA until the elongation is done. And so in the, to the left, we have a dimer form of DNA polymerase that is present in prokaryotic cells. And so the eukaryotic example is in a trimer form. And so there's homology between them, but rather the way in which the subunits come together. This goes back to your question, Debbie, whether there's a difference between the DNA polymerase and prokaryote versus the eukaryote. They're the same, but it's just a matter of how those subunits are coming together that can distinguish them. And so this the takeaway the takeaway from this slide is to try and illustrate how this actual structure helps to ensure that DNA polymerase 3 or DNA pol delta is able to hold on and not let go until elongation is done during DNA replication. All right, I think I'm going to go ahead and hit pause there. And so, are there any questions on any of this so far? This is a lot. Yes. Yeah. So the main takeaway, or the main take-home message for the processivity clamp of DNA polymerase is that it is clamping down and holding on to the template DNA during elongation, and it does not let go until elongation is completed. And so it's not this is not a chemical feature of DNA polymerase, but rather just a physical feature that enables it, based on its structure, to complete elongation. Because when we talk about, historically when we've talked about, this is the important point I missed. When we've talked about enzymes, we've described how every enzyme has a substrate, and we have enzyme substrate, then we have the enzyme substrate complex, and then once we have a product, the enzyme lets go, and so now it's going back to its substrate. With DNA polymerase, it's not letting go, and it's able to continue mediating its enzymatic reaction until this entire process of elongation is done. And so this is meant to address how enzymes typically behave, but in this case, it's not letting go because it's meant to go through the entire process of elongation until replication is done. So to try and summarize so far what we've talked about, just in a big picture, we talked about how DNA replication, the sites where it happens are these replications of origin. Bacteria or prokaryotes have a single replication origin site. We have tens of thousands. We have these initiator proteins that come in and bind to those replication origin sites. We have helicases that will open up and melt the DNA. We have the topoisomerases that help alleviate the supercoiling. And we have the DNA polymerase that comes in and helps mediate the elongation. There are a couple more pieces of the puzzle that we're going to talk about in the next lecture, but what will help is to try and map out those like what each protein complex or each protein does during this process. Okay. If you have any questions, I'll be waiting up here, and thanks for giving me your time.
I just wanted
Sorry,
to make
give me
a
1 second. Let me go ahead and end this real quick.